Today we are discussing our next topic, chromatin and its effects on DNA metabolism. Firstly, we have to come to know about the chromatin. Chromatin is nothing but that is the early form of the chromosome, and this chromatin that will be found in the fibrosperm or thread-like structure. On this chromatin, there is the situation of the DNA, and there is the specific type of the arrangement that will be present of the DNA on the chromatin. This chromatin directly affect on the metabolism of the DNA, and in this metabolism, there will be including the transcription. After that, the arrangement of the DNA on the chromosome, and this chromatin also regulate the different type of the functions of DNA. The chromatin is the material that make up the chromosome that consists of DNA and protein. Mostly these chromatin fibers that will be form the chromosome, and this chromosome that will be consist of two components. First component is the DNA, and the second component is the protein that wrapping or that surround the particularly DNA. This protein that is the particularly basic protein, and the name of this protein is the histone protein. The major protein in chromatin are protein called the histones. Means in the particularly chromatin, the DNA that will be surrounded by the protein or DNA that will be remain in contact with the protein, and this protein is known as the histone protein. This protein that will act as a packaging element for the DNA. Means this protein that will be surround the particularly DNA that will be situated on the chromosome. The reason that the chromatin is important is that it is very pretty good packaging trick to get all the DNA inside a cell. And this chromatin that will be or this protein that will be present on the chromatin that wrap the DNA or that surround the DNA, and so that this packaging of this particularly DNA by the chromatin that will be enable the DNA that will be remain inside the particularly cell. Means the DNA that will be remain in the nucleus in the wrap condition or in the particularly packed condition due to the presence of this protein, and this protein is the histone protein. These chromatin fibers are not condensed but can exist in either a compact form. म्हणजे this chromatin fiber or the chromatin only there will be the situation of the DNA that will be basically divided into the particularly two types. The compact form of the chromatin that is known as the heterochromatin, or less compact form of the chromatin that is known as the euchromatin. Means basically this chromatin fiber on which the DNA is situated depending upon their particularly structure, either that will be compact or the particularly loose. When the chromatin is compact, that will be the heterochromatin, and when the chromatin is less compact, that is known as the euchromatin. Processes including DNA replication, transcription, and combination occur in euchromatin. Mostly, this particularly DNA or the different that is the particularly function of the DNA that will be including the DNA replication, म्हणजे formation of the duplicate copy of the DNA. After that, transcription means formation of the messenger RNA from the DNA, and recombination. Recombination means there is the particularly combination of the two DNA that will be mostly occur in the euchromatin, which will be the particularly which will be the particularly that is the loose or that is the less compact type of the protein. During cell division, chromatin contains to form the chromosome, and this chromatin or the early that is the chromosome that is known as the chromatin, and this chromatin that will be formed the particularly chromosome or densely condensed form of the chromatin that is known as the chromosome. Means from that we can say that chromosome is nothing but that is a condensed form of this particularly. Condensed means that will be most thickened, and there will be the particularly coming the chromatin at the single point. Chromatin does not only determine gene expression and epigenetic integrity of the nuclear DNA. म्हणजे chromatin not only determine the gene expression, म्हणजे expression of the gene or giving of the character of the gene, and epigenetic integrity. Epigenetic integrity means this the effect of the environment on the particularly structure of this particularly DNA, that is known as the epigenetic integrity of nuclear DNA, but directly contribute to the repair of genomic lesion. But this particularly chromatin that will be related with the directly on the particularly repair of the genomic lesions, means repairing of the small break of the particularly genome or the particularly DNA that will be present on the haploid set of chromosome. The small that is the number of this particularly damage, and this damage that will be repaired by means of this particularly chromatin of the DNA double stranded break. And in this particularly damage, there will be including of the DNA double stranded break, means breaking of the double stranded DNA into the small fragment are the most toxic, and such type of the breaking of the double stranded DNA that is very harmful, or particularly most harmful to the particularly functioning of the DNA. The need for chromatin reorganization during eukaryotic DNA repair. The need of chromatin reorganization means there will be requirement of the chromatin reorganization, means there will be the structural that is the particularly reorganization during eukaryotic DNA repair, that will be occur in the particularly DNA repair in the eukaryotic DNA. Eukaryotic DNA means the DNA of the that is the nucleated cell, and it become appear in light of the tight packaging of the DNA in nuclear space, and this chromatin that will be helpful to the particularly light of the tight packaging, when the light of the tight packaging of the DNA in nuclear space, and this chromatin that will help the DNA for the packaging or tight packaging of the DNA. In human and mice, this type of the condition that will be occur. Approximately every 200 base pair, 146 to 147 base pair of DNA are wrapped around the nucleosome, and it will be noticed that during this packaging there will be approximately 200 base pair, म्हणजे 200 base that will be paired with the complementary nitrogen base, or 146 to 147 base pair of DNA, and sometimes it will be noticed that 146 to 147 base pair of the DNA are wrapped around the nucleosome. Are wrapped means that will be surrounded around the nucleosome. What is the nucleosome? Nucleosome is nothing but that is the particularly small bead like structure that will be present on the particularly chromatin, and this bead that will be containing the octamer of the particularly that is the histone protein, and this histone protein that will be basically divided into different types, that is the H2A, H2B, H3, H4. These are the four histone proteins that will be found in the dimer form, and so that there will be the total eight types of this particularly histone protein that will be wrapped, and that form the bead-like structure, which form the structural core of the chromatin fiber. And this particularly wrapping, or there is the accumulation of this particularly base that is the histone protein that will be useful for the particularly wrapping of the particularly chromatin fiber, or there will be accumulation of the chromatin fiber. This is the structure of this particularly DNA, and there is the relation with the particularly chromatin fiber. This is the DNA, and here there will be the formation. There will be the formation of the double-stranded DNA, and also there will be seeing the double. That there is the formation of the complementary nitrogen base pairing. Here, you find that C that will be attached with the G, or there is the pairing of the C with the G, and T that will be with the particularly A. So in this way, there will be the double stranded DNA here will be found. But in this case, you see that there will be the 4 unit at the particularly upper side and 4 unit that will be particularly lower side. So in this 4 unit, there will be containing the histone protein. Basically, histone protein that will be divided into 5 types. That is the H1, H2A, H2B, H3 and H4. Out of that, this H1 that will be always present outside this particularly this octamer, and that will be present, or that will be remain attached to the linker DNA. This is the linker DNA, and this DNA that will be wrapping around this particularly octamer. After that, again there is the linker DNA, and again there is the next octamer that will be found. Octamers means there is a 8 unit of the histone protein, and in this 8 unit there will be again there is the presence of 4, that is the 4 unit that will be upper side and 4 unit at the lower side. In this 4 unit there will be containing the H2A, H2B, H3 and H4. Again there will be the same amount of the histone protein that will be present on the lower side, that is the H2A, H2B, H3 and H4. And these two that is the octamer that will be joined by the specific DNA that is known as the linker DNA. And to this linker DNA, here you can see that in this particularly case, there will be presence of this particularly single unit of the histone protein, and this is the H1 protein. This is the H1 or H1 histone protein, and always H1 histone protein that will be remain attached to the particularly linker DNA. This is the linker DNA, and to this linker DNA, H1 protein is present. Here also H1 protein is present, but this for this structure that will be showing the octamer. Octamer means there will be contained the 8 unit in the particularly dimer form of the H2A, H2B, H3 and H4. In this way, there will be the structure. This is also the particularly chromatin, and this chromatin is the compact type of the chromatin, and this compact type of the chromatin that will be known as the particularly heterochromatin. This is the heterochromatin, and this heterochromatin that will be not particularly found in the process of the transcription or in the particularly formation of the messenger RNA in the protein synthesis. So in this way, when you see the structure of this particularly chromatin, there is the relation of the chromatin with the particularly DNA, and there is the different type of the unit that will be also seen, like the particularly histone protein is also seen in the structure of the DNA, and this combined structure that is the chromatin, that is the histone protein, and the DNA that form the particularly complete, that is the chromosomal structure. Each nucleosome consists of the histone octamer. I have said that each nucleosome that is nothing but that is the particularly bead-like structure, and this bead-like structure that will be formed by the particularly octamers of different type of the particularly histone protein, and in each octamer there will be containing the two molecules of H2A, H2B, H3 and H4. So this H2A, H2B that is the particularly histone protein, H3 is another histone protein, and H4 is another histone protein. So these all four molecules that will be found in two form, and so that there is the formation of the octamer or variant thereof assembled in one S3 and S4 heterochromatin, assemble in one S3 S4 heterotetramer and two H2A and H2B heterodimer. तर here what actually happen, S3 and S4 assemble in one particularly heterotetramer, and two H2A H2B that is the heterodimer, because these are the H2A and H2B that will be present in dimer form. Dimer form means equal, that is the same form but particularly in two group. But this particularly S3 and H4, म्हणजे histone protein S3 and H4 always found in the particularly heterotetramer. Heterotetramer means that will be different. Nucleosomes are subjected to further condensation. Nucleosome that will be particularly again that will go into the condensation. Condensation means that is the particularly deeply that is the that will be a close association with each other, the H1 linker, and that will be take place by means of the linker DNA, linker DNA that will be containing the H1. So you see in the particularly diagram, it will be noticed that this octamer that will be always found in the condensed form, in the closely associated form, and this close association of the octamer that will be particularly take place by means of the particularly linker DNA. This is the linker DNA, and on this linker DNA there will be the presence of the H1 histone. H1 histone means H1 histone protein. So this H1 histone protein indirectly bring the octamer in compact form by the tightening of this particularly DNA, and so that when you see or resemble this particularly structure, this structure that will be look like the sting or the particularly beads. This is the sting and this is the bead, and the condition is nothing but that is the beads are woven in the sting. Beads are woven in the sting. These are the particularly beads, and that will be woven in the particularly sting. And sting is nothing but that is the linker DNA. The sting is the linker DNA, and this bead is nothing but that is the octamer of the particularly histone protein. So in this way, this compactness of the octamer that will be occur or that will be occur due to the particularly compact arrangement of the octamer by means of the particularly linker DNA, and this for this compactness there is the help of this particularly histone protein or H1 protein, H1 histone protein. So this structure that will be seen. Nucleosomes are subjected to further condensation via the H1 linker histone, which contact the exit entry point of the DNA. So this H1 linker histone or H1, that is the protein which contact or which will remain in contact with the particularly exit entry point of the DNA. म्हणजे exit and entry point of the DNA, there will be the position of the H1 linker, DNA stand on the nucleosome and facilitate the higher order chromatin folding. म्हणजे this folding of the particularly chromatin this chromatin folding, that will be dependent on the particularly H1 linker DNA, and this H1 linker DNA that will be present on the exit and entry point. You see here there will be the H1, this H1 that will be present at the particularly end of this particular DNA, and that is the exit point, or that will be present before the particularly this nucleosome or the octamer, that is known as the particularly entry point. So this entry point and exit point that will be occupied by the linker DNA, and this linker DNA that will be remain attached with the H1 protein. The position of the H1 protein always present at the particularly entry point and the exit point, and this will be helpful for the particularly that is the particularly folding of the chromatin fiber. म्हणजे the folding or higher order of chromatin folding, that will be dependent on the particularly this H1 protein, and that will be helpful for the compactness of the nucleosome. While the most prevalent epigenetic alteration of DNA, when there is the epigenetic means a certain type of the environmental factor that will be change the structure of the DNA or methyl base modification of the cysteine, and this type of the changes or the alteration of the particularly DNA by means of the epigenetic way. Epigenetic means by means of the environmental, that is the factor, and in that alteration there is the inclusion of the methylation or methyl base modification, or there is the addition of the methyl group in the particularly DNA, or sometimes there is the removal of the methyl group of cysteine in CpG context. म्हणजे this methyl group that will be removed from the cysteine, cytosine like the cytosine nitrogen bases. Histones are subject to a wide range of reversible. आता these histones that will be bring the particularly changes, and these changes are reversible changes. Reversible changes means the changes that will be particularly bring adversely, and again the same changes that will be bring, that is the particularly favorably, and this type of the changes that is known as the reversible changes. And these reversible changes that will be bring by means of this particularly histone protein in the structure of the DNA. Most transcriptional modification, post-transcriptional modification, including the acetylation, methylation or phosphorylation, ubiquinylation or sumolylation, and ADP ribosylation. आता there are the different type of the post-transcriptional changes. Post-transcriptional changes means the changes that will be take place in the particularly DNA, or the changes that will be take place in the particularly RNA after the particularly formation of the messenger RNA. Post transcription म्हटलं त्याने. Post transcription म्हणजे when the DNA that form the messenger RNA, and after the formation of the messenger RNA, the changes that will be or the modification that will be occur in the particularly DNA, म्हणजे transcriptional DNA किंवा the DNA that will be doing the transcription, or sometime the messenger RNA that will be formed from the particularly DNA, and that particularly messenger RNA, the changes that will be mostly the type of the acetylation. Acetylation means there is the formation of this particularly acetyl group. Sometimes there is the changing of the methylation, means there is the addition of the methylation group or changing of the methyl group, CH group. Sometimes there will be the particularly changes of the phosphorylation, means there is the change in the that is the phosphate group, or sometimes again there is the particularly changes in ADP ribosylation means ADP in adenosine diphosphate ribosylation. So these all changes that are the post transcriptional changes, म्हणजे the changes of nature like the particularly acetylation, methylation, phosphorylation, or ubiquinylation, or ADP, that is a ribosylation, So these all changes that will be the post-transcriptional changes, and these post-transcriptional changes that will be bring or the comes after the transcription of the DNA, or when the DNA from the particularly messenger RNA, and so that these post-transcriptional changes that will be particularly bring the changes in the formation of the protein synthesis. म्हणजे whatever the nitrogen bases that will be present on the particularly parent DNA. Parent DNA means the DNA that will be particularly from the messenger RNA. But whatever the nitrogen bases present on the particularly double stranded DNA, and whatever the nitrogen bases that will be given in the particularly messenger RNA, that will be totally different. And this type of the changes, म्हणजे always whatever the nitrogen bases that will present on the double stranded DNA, and same that is the complementary nitrogen bases that will be come into the particularly messenger RNA. But when you compare the nitrogen bases on the parent DNA which will be from the messenger RNA, that will be not exactly similar to the particularly nitrogen bases that will come into the messenger RNA, and such type of the changes in the nitrogen bases in the transcription that will be come under the heading of the post transcriptional modification, म्हणजे the changes that will be bring after the particularly transcription. And transcription is the process in which the double stranded DNA that will be give the single stranded messenger RNA. While the most prevalent epigenetic alteration of DNA are methyl based modification of cytosine, uh, cytosine content, histones are subjected to a wide range of reversible and post transcriptional changes that will be also the particularly same type. Changes in metabolism and consequently metabolism, change in the metabolism and consequently metabolite availability are tightly associated with both aging and cancer, raising the intriguing possibility that age disease associated. आता most of the changes that will be bring in the particularly metabolism of the DNA by means of the particularly chromatin fiber, तर these changes that will be associated with both type of this particularly condition. Either this particularly changes that will bring by means of the aging. Aging means when the cell that will be undergoes the period of the particularly aging, and due to aging there is the structural and functional modification that will be take place in the cell, and by means of the aging most of the particularly metabolic changes in the DNA that will bring, and for that particularly responsible factor is the chromatin. Sometimes these changes that will be occur due to the particularly cancer that will be also that will be also responsible for the bringing of the changes in the metabolism of the DNA and raising the inter the intriguing possibility that age or disease associated metabolic alteration, and such type of the changes that will be bring in the particularly DNA, or such type of the metabolic changes that will occur in the DNA, that will be associated with particular disease associated metabolic alteration and can result in the deregulation of DNA repair by altering the chromatin function. And due to this particularly cancer or the aging, whatever the changes that will be occur in the DNA, or whatever the particularly damaging that will be occur in the DNA, and this DNA damaging that will be particularly not repair, or that will be particularly not repaired by means of the particularly chromatin. म्हणजे mostly this chromatin that will be repair the different type of the changes bring in the DNA, because this chromatin that will be always remain in contact with the DNA. But sometime it will be noticed that whatever the changes that will be related with the aging, or whatever the changes that will be related with the cancer disease, and that bring the particularly breakage or the particularly damage in the DNA, and such type of the damage that will be not repaired by means of the particularly chromatin, and that will be due to the altering of the particularly chromatin, that is the function. म्हणजे there is the changing of the chromatin function, or by means of this particularly aging and cancer, the chromatin function that will be changed, and due to the changing of the chromatin function, the aging and cancer or aging and the disease like cancer that bring the changes in the particular structure of the DNA, and these changes include the damaging the certain part of the DNA, and this damaged part that will be not repaired by the particularly chromatin due to the changing of the chromatin function, or chromatin that will be not remain in normal condition. The normal functioning of the chromatin is nothing but that is the repairing of this particularly DNA, or they whatever the change that will be bring in the DNA that will be repaired. But this in this condition, that is the aging and cancer condition, whatever the breakage or what is the damage that will occur in the DNA, that will be not repaired by this particularly this chromatin, and that will be due to the changing of the chromatin function. This is the end of. After that, firstly you see this again this you see the structure. This is the particularly structure of the nucleosome solenoid model, or this type of the DNA structure is also known as the nucleosome solenoid model. याला आपण nucleosome solenoid model पण म्हणू. आता याच्यामध्ये काय आहे बघा, here there will be containing the this is the DNA, and this DNA is known as the linker DNA. Linker DNA means the DNA that will be present, that will be outside the nucleosome, that is not wrapping to the particularly nucleosome. And what is the nucleosome? Nucleosome is nothing but that is the particularly octamer of the histone protein. आता हा जो octamer दिसते, this is one octamer, this is second octamer, this is third, and this is fourth. But here there is no octamer. There is the only single that is the particularly histone that will be seen. So what actually happened? Generally, this structure is the particularly linker DNA, and this linker DNA that will be made up by this particularly double stranded DNA. This is the double stranded DNA, and in this double stranded DNA there is a complementary nitrogen base pairing that will be seen. That is the complementary base pairing that will be seen. At this complementary base pairing, there will be containing the pairing of the A adenine to the thymine, cytosine to the guanine, and guanine to the cytosine. Means normal structure of this particular DNA. But what happened? When this DNA that will be going here, that is from the double stranded DNA, in this particular structure, there is the wrapping of the DNA, or there is the packaging of the DNA to the particularly octamer. म्हणजे this octamer that will become compact, or all the unit of the octamer. Mostly in the octamer, which type of the units are present? There will be containing the basic protein or the particularly histoprotein in the form of this particularly 4 unit. So in this upper 4 unit, there will be containing the H2A, H2B, H3, and H4, and below again the same unit. And so that this 4 unit and this 4 unit that will form the octamer. So this compactness of this particularly octamer that will depend upon the DNA packaging. Means how this particularly DNA pack this octamer. When there is a tightly packed, that time this octamer that will become the tight. So this tightly packaging depending upon this particularly type of the chromatin. That type of the chromatin means chromatin that will be the U type of the chromatin. That time there is no tightly packing. But when the chromatin that is the heterochromatin, that will be tightly packing. Again, you know that U chromatin have the genetical importance, because U chromatin always involved in the particularly transcription. But heterochromatin that will be not always involved in the transcription, and so that tightly packing that will be seen here. Here there is a tightly packing. Tightly packing means there is the compactness, more compactness of the particularly histone protein found here, and this tightly packing of this particularly this octamer that will be due to the heterochromatin. But in this particularly case, there is the loose packing, because there is the individual or the individual octamer that will be seen, and this individual octamer that will be seen, that will be due to the loose packing. Loose packing means here there is the involvement of the U chromatin, and U chromatin have the particularly genetic importance, because U chromatin that will be always involved in the transcription. आता what actually happen when there is the alteration or the change? म्हणजे most of the metabolism or most of the metabolism of the DNA that will be dependent upon the particularly chromatin, and this chromatin that will be involved in the particularly packaging or the particularly repairing of the particularly damaged part of the DNA. So what actually happen when there is the damaged part of the DNA that will be found in the particularly DNA strand? That time, depending upon the packaging, This chromatin that will be take part in the, that is in the particularly curing of the package, curing of the damaging, or there is the particularly repairing of the particularly DNA. Repairing of the DNA means the repairing of the damaged part of the DNA. Again, it will be noticed that due to the some epigenetic, that is a factor. Epigenetic means some time of the unknown or the particularly environmental factor. So this unknown and environmental factor that will be damage the particularly DNA, that will be damage the DNA, and this damaging of the DNA that will be tagged or that will be particularly cured by means of the chromatin, because there is a direct role of the chromatin on the particularly, there is the direct role of the chromatin on the DNA repair or the metabolism of the DNA. म्हटलं त्यांनी metabolism of the DNA म्हणजे breaking of the DNA or the joining of the DNA, and breaking of the DNA and joining of the DNA, there will be the role of this chromatin. After that, he will, or it will be also said that the two type of the condition, that is the particularly sometime there will be the epigenetic factor or aging and the particularly cancer. आता aging means what actually happen when the cell that will be become, that is the particularly old, and due to oldness of the cell or due to the particularly oldness of the cell, some type of the physical and physiological or structural changes that will be take place in the cell, and cell that will be lose their original, that is the particularly structure. So this aging, that is also the one of the factor that will be bring the changes or the damages in the particularly DNA, and this DNA damaging due to the aging that will be not cured by the particularly chromatin, because due to the aging again there is a altering the function of this particularly chromatin, which a chromatin that will not remain in the particularly that is the same intensively, or that will be not showing the same intensity. Means the normal function of the chromatin is the repairing of the DNA or whatever the changes that will be bring in the DNA, that will be cured by the this particularly chromatin. But due to the aging, whatever the changes that will be bring in the DNA, that will be not cured by the chromatin, and that is due to the particularly alteration or the changing of the function of the chromatin fiber. Such type of the mechanism also seen in the particularly cancer cases. In the cancer cases, again there is a certain time of the damage or the particular incision म्हणू आपण त्याला. Incision म्हणजे small, that is the breakage of the double stranded DNA or the single stranded DNA. एका स्टँडला असेल किंवा दोन स्टँडला असेल ब्रेक, तर that will bring the particularly cancer. Cancer bring these particular changes, and again these changes that will be not particularly cured by the chromatin. So from this it will be clear that there is the role of the chromatin fiber on the particularly metabolism of the DNA. Metabolism of the DNA म्हणजे the break of the DNA or the joining of the DNA. There is the role of this particularly chromatin, and chromatin that will be mostly present in the particularly two form, that is the euchromatin and the heterochromatin. Euchromatin, that is the particularly looseness or less compact type of the chromatin, and mostly involved in the particularly transcription process. But hetero type of the chromatin, that is the condensed, that is the particularly compactness or compact type of the chromatin, or deeply, that is the that will be also like a stain deeply. Heterochromatin that will be also stain deeply, and that will be present in the particularly compactness, and this euchromatin that will be less compact. So this euchromatin have the role in the particularly formation of the messenger RNA or transcription, and this heterochromatin that will be no role in the particularly transcription. And genetical point of view, when you see the genetical point of view, the euchromatin is helpful as compared to the heterochromatin, and always this particularly chromatin that will be remain in the particularly chromosome. And when in the during the particularly metaphase of the cell division, when this chromatin that will become condensed and thick, and this form of the chromatin that is known as the chromosome. So this chromosome is nothing but that is a condensed form of this particularly chromatin, and there is the role of this particularly chromatin in the metabolism of the DNA. Means in the for the different type of the functioning of the DNA, that will be there will be involvement of this particularly chromatin. But whatever the changes that will be bring, or that is the epigenetic changes, mean the changes that will be by means of the certain type of the chemical factor or certain type of the particularly environmental factor, there will be the changing of the actual composition of this particularly DNA or the particularly chromatinous म्हणू आपण किंवा DNA म्हणू, तर that will be mostly of acetylation, methylation, phosphorylation, means there is a changing of the different type of the particularly compound, and these changes that will be brought or that will be bring by means of the epigenetic factor or environmental factor, or sometime due to the addition or the due to the particularly addition of the chemical. So in this way there will be the role of this particularly chromatin, and there is an effect of the chromatin on the DNA metabolism. So I think that this point that should be well understand to you, and you got this point. I hope that you get this point very well and you can explain this. Okay.